Section 20 of Gray's Anatomy, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information, or to volunteer, please visit LibriVox.org. Recording by Mark Roche. 2G. The Small Intestine. Structure. The wall of the small intestine is composed of 4 coats: serous, muscular, areolar, and mucous. The serous coat, tunica serosa, is derived from the peritoneum. The superior portion of the duodenum is almost completely surrounded by this membrane near its pyloric end, but is only covered in front and at the other extremity. The descending portion is covered by it in front, except where it is carried off by the transverse colon, and the inferior portion lies behind the peritoneum, which passes over it without being closely incorporated with the other coats of this part of the intestine, and is separated from it in and near the middle line by the superior mesenteric vessels. The rest of the small intestine is surrounded by the peritoneum, excepting along its attached or mesenteric border. Here a space is left for the vessels and nerves to pass to the gut. The muscular coat, tunica muscularis, consists of 2 layers of unstriped fibers, an external longitudinal and an internal circular layer. The longitudinal fibers are thinly scattered over the surface of the intestine, and are more distinct along its free border. The circular fibers form a thick, uniform layer, and are composed of plain muscle cells of considerable length. The muscular coat is thicker at the upper than at the lower part of the small intestine. The areolar or submucous coat, tela submucosa, connects together the mucous and muscular layers. It consists of loose filamentous areolar tissue containing blood vessels, lymphatics, and nerves. It is the strongest layer of the intestine. The mucous membrane, tunica mucosa, is thick and highly vascular at the upper part of the small intestine, but somewhat paler and thinner below. It consists of the following structures. Next, the areolar or submucous coat is a double layer of unstriped muscular fibers, outer longitudinal and inner circular. The muscularis mucosae internal to this is a quantity of retiform tissue, enclosing in its meshes lymph corpuscles, and in this the blood vessels and nerves ramify. Lastly, a basement membrane supporting a single layer of epithelial cells, which throughout the intestine are columnar in character. The cells are granular in appearance, and each possesses a clear, oval nucleus. At their superficial or unattached ends, they present a distinct layer of highly refracting material, marked by vertical striae, the striated border. The mucous membrane presents for examination the following structures contained within it or belonging to it: circular folds, villi, intestinal glands, duodenal glands, solitary lymphatic nodules, aggregated lymphatic nodules. The circular folds, plicae circulares, Kerkringi, valvuli conniventes, valves of Kerkring, are large valvular flaps projecting into the lumen of the bowel. They are composed of reduplications of the mucous membrane, the 2 layers of the fold being bound together by submucous tissue. Unlike the folds in the stomach, they are permanent, and are not obliterated when the intestine is distended. The majority extend transversely around the cylinder of the intestine for about 1/2 or 2/3 of its circumference, but some form complete circles, and others have a spiral direction. The latter usually extend a little more than once around the bowel, but occasionally 2 or 3 times. The larger folds are about 8 mm in depth at their broadest part, but the greater number are of smaller size. The larger and smaller folds alternate with each other. They are not found at the commencement of the duodenum, but begin to appear about 2.5 or 5 cm beyond the pylorus. In the lower part of the descending portion, below the point where the bile and pancreatic ducts enter the intestine, they are very large and closely approximated. In the horizontal and ascending portions of the duodenum and upper half of the jejunum, they are large and numerous, but from this point down to the middle of the ileum they diminish considerably in size. In the lower part of the ileum they almost entirely disappear. hence the comparative thinness of this portion of the intestine as compared with the duodenum and jejunum. The circular folds retard the passage of the food along the intestines, and afford an increased surface for absorption. The intestinal villi, villi intestinales, are highly vascular processes, projecting from the mucous membrane of the small intestine throughout its whole extent, and giving to its surface a velvety appearance. They are largest and most numerous in the duodenum and jejunum, and become fewer and smaller in the ileum. Structure of the villi. The essential parts of a villus are the lacteal vessel, the blood vessels, the epithelium, the basement membrane, and the muscular tissue of the mucosa, all being supported and held together by retiform lymphoid tissue. The lacteals are in some cases double, and in some animals multiple, but usually there is a single vessel. Situated in the axis of the villus, each commences by dilated cecal extremities near to, but not quite at, the summit of the villus. The walls are composed of a single layer of endothelial cells. The muscular fibers are derived from the muscularis mucosae, and are arranged in longitudinal bundles around the lacteal vessel, extending from the base to the summit of the villus, and giving off, laterally, individual muscle cells, which are enclosed by the reticulum, and by it are attached to the basement membrane and to the lacteal. The blood vessels form a plexus under the basement membrane, and are enclosed in the reticular tissue. These structures are surrounded by the basement membrane, which is made up of a stratum of endothelial cells, and upon this is placed a layer of columnar epithelium, the characteristics of which have been described. The retiform tissue forms a network in the meshes of which a number of leukocytes are found. The intestinal glands, glandulae intestinales libercunae, crypts of Lieberkühn, are found in considerable numbers over every part of the mucous membrane of the small intestine. They consist of minute tubular depressions of the mucous membrane, arranged perpendicularly to the surface, upon which they open by small circular apertures. They may be seen with the aid of a lens, their orifices appearing as minute dots scattered between the villi. Their walls are thin, consisting of a basement membrane lined by columnar epithelium, and covered on their exterior by capillary vessels. The duodenal glands, glandulae duodenales Bruneri, Brunner's glands, are limited to the duodenum, and are found in the submucous areolar tissue. They are largest and most numerous near the pylorus, forming an almost complete layer in the superior portion and upper half of the descending portions of the duodenum. They then begin to diminish in number and practically disappear at the junction of the duodenum and jejunum. They are small compound acinotubular glands consisting of a number of alveoli lined by short columnar epithelium and opening by a single duct on the inner surface of the intestine. The solitary lymphatic nodules, noduli lymphaticae solitarii, solitary glands, are found scattered throughout the mucous membrane of the small intestine, but are most numerous in the lower part of the ileum. Their free surfaces are covered with rudimentary villi, except at the summits, and each gland is surrounded by the openings of the intestinal glands. Each consists of a dense interlacing retiform tissue closely packed with lymph corpuscles, and permeated with an abundant capillary network. The interspaces of the retiform tissue are continuous with larger lymph spaces which surround the gland, through which they communicate with the lacteal system. They are situated partly in the submucous tissue, partly in the mucous membrane, where they form slight projections of its epithelial layer. The aggregated lymphatic nodules, noduli lymphaticae aggregatae, Peyer's patches, Peyer's glands, agminated follicles, tonsillae intestinales, form circular or oval patches, from 20 to 30 in number, and varying in length from 2 to 10 centimeters. They are largest and most numerous in the ileum. In the lower part of the jejunum they are small, circular, and few in number. They are occasionally seen in the duodenum. They are placed lengthwise in the intestine, and are situated in the portion of the tube most distant from the attachment of the mesentery. Each patch is formed